Hello and welcome to the Midnight Lab. Before we begin tonight's journey, take a moment to like the video and subscribe to the channel if you enjoy calm explorations of science and the universe. And while you're settling in, I'd love to know, where in the world are you watching or listening from tonight? Maybe you're listening from a quiet apartment in a city that never fully sleeps. Maybe you're somewhere rural, where the night is deeper and the stars feel closer. Or maybe you're halfway across the world from someone else watching this same video at this exact moment. Wherever you are, the body you're listening with right now carries a story that stretches back millions of years. The shape of your spine, the structure of your hands, the way your eyes adjust to the dim light around you, even the quiet rhythm of your breathing. None of these things appeared suddenly. Every bone, every muscle, every organ is the result of a long chain of small changes, shaped by survival, environment, and chance across vast stretches of time. In a sense, your body is a living archive. Inside it are traces of ancient forests where early primates once leapt between branches, clues from open African grasslands where our ancestors first stood upright, echoes of long-distance journeys, shifting climates, and countless generations adapting to the world around them. Evolution did not design the human body all at once. Instead, it modified what already existed, reshaping ancient biological structures into new forms, often solving new problems with old tools, which is why the human body is full of strange compromises. A spine built for walking upright, but prone to back pain, teeth that once handled tougher diets, but now often need removing, a brain powerful enough for language, imagination and science, yet one that consumes an enormous share of our body's energy. These are not flaws, they are footprints of our evolutionary past. And tonight we're going to follow those footprints. We'll explore how shifting climates helped shape the way we walk, how ancient diets influenced the size of our brains, why our skin, muscles and senses evolved the way they did, and how even the smallest features of the human body can reveal clues about the environments our ancestors once lived in. Along the way, we'll encounter the ideas of scientists, anthropologists, and evolutionary biologists who have spent decades piecing together this story from fossils, genetics, and the quiet clues hidden within our anatomy. Because the remarkable truth is this: you don't need a time machine to study human evolution. You only need to look closely at the body you already have. So tonight, as the world around you grows quieter, we're going to step back through deep time and explore the long evolutionary journey that shaped the human body. From ancient primates in the trees, to early humans walking across African landscapes, to the complex biological system you inhabit today. This is the story of how evolution shaped the human body. Let's begin. If you take a moment to notice your own body right now, something remarkable becomes clear. The shape of your hands resting in your lap, the quiet movement of your lungs as you breathe, the steady beat of your heart deep in your chest. All of it feels immediate and present, part of the ordinary experience of being alive, and yet every part of that experience is the result of a story that began unimaginably long ago. The human body is not a finished design that appeared suddenly in the world. It is better understood as an evolving system, assembled gradually over immense stretches of time as living organisms adapted to the environments around them. Every bone, every muscle, every organ has been shaped by a combination of environmental pressures, genetic variation, and the quiet but persistent force of natural selection. The idea that species change over time was not always widely accepted. In the middle of the 19th century, two naturalists working independently arrived at a similar realization. Charles Darwin and Alfred Russel Wallace both proposed that species evolve through a process they called natural selection. In simple terms, individuals within a population vary in small ways. Some of those variations help them survive and reproduce more successfully in a particular environment. Over many generations, those advantageous traits become more common. Darwin famously described this process in his book On the Origin of Species, published in the year 1859. Wallace reached similar conclusions while studying the wildlife of the Malay archipelago. Their shared insight changed biology forever. Instead of viewing living creatures as static forms, scientists began to see them as populations constantly shaped by the pressures of survival. Natural selection works slowly, often so gradually that its effects are almost impossible to notice within a single human lifetime. But given thousands or millions of years, Even tiny differences can transform an organism in profound ways. A slightly longer limb, a small shift in metabolism, a minor change in bone structure. Over generations, these small adjustments accumulate, gradually producing entirely new anatomical patterns. In the case of the human body, this process has unfolded across several million years of evolutionary history. Our anatomy carries the marks of ancient environments, long vanished landscapes, and survival challenges faced by countless ancestors. But Darwin and Wallace were only the beginning of the scientific story. In the early 20th century, biologists began to connect Darwin's ideas with the emerging science of genetics. This intellectual framework became known as the modern evolutionary synthesis. One of the key figures in developing this synthesis was evolutionary biologist Ernst Mayr. Mayr helped clarify how genetic variation within populations fuels evolution. Genes are inherited units of information that influence traits such as body structure, physiology, and behavior. Mutations introduce new variations into those genes. Most mutations have little effect, and some can be harmful. But occasionally a mutation produces a change that improves survival or reproduction in a specific environment. When that happens, natural selection can gradually spread that genetic variant through a population. Over long periods of time, these genetic shifts accumulate and reshape species. The modern evolutionary synthesis united several scientific fields including genetics, paleontology, systematics, and population biology. It showed that evolution is not simply a vague historical idea, but a measurable biological process operating through the interaction of genes, variation, and selection. To visualize how populations evolve, geneticist Sewall Wright introduced an idea that has become one of the most influential metaphors in evolutionary biology. Wright described what he called an adaptive landscape. Imagine a vast landscape made not of hills and valleys of rock and soil, but of peaks and valleys representing survival success. Each peak represents a combination of traits that allows organisms to survive and reproduce effectively in a given environment. Each valley represents less successful combinations. A population in this metaphor moves across the landscape over time. Genetic variation and natural selection push it gradually uphill toward higher peaks of adaptation. But environments change, and new peaks can appear while old ones disappear. As conditions shift, populations must adapt again, exploring new evolutionary paths across this ever-changing terrain. The human body is one such path through the adaptive landscape. Our anatomy represents a particular set of evolutionary solutions shaped by ancient climates, food sources, predators, and social interactions. Yet even this picture is not complete. More recently, some scientists have suggested that evolution cannot always be understood by focusing on genes alone. One of the researchers who has argued strongly for a broader perspective is systems biologist Dennis Noble. Noble and other scientists in systems biology emphasize that living organisms are deeply interconnected systems. Genes influence cells, cells influence tissues, tissues influence organs, and organs interact within entire physiological networks. Feedback loops operate at many levels of the body. For example, the development of bone is not controlled by genes alone. Mechanical forces produced by movement also influence bone density and structure. Hormones regulate metabolism in ways that affect growth and development. Environmental conditions interact with biological systems in complex ways. From this perspective, evolution shapes not just individual traits, but entire networks of biological interactions. A change in one system often affects several others. Over time, natural selection operates on the performance of the whole organism. This helps explain why the human body often appears both remarkably capable and strangely imperfect at the same time. Our spine, for instance, allows us to walk upright and free our hands for complex tasks, but that same structure is prone to lower back pain because it evolved from a spinal design originally suited for four-legged movement. Our teeth once served ancestors who consumed tougher diets of raw plants and meat. In the modern world, those same teeth sometimes crowd together uncomfortably in jaws that have gradually become smaller. These features are not signs of poor design. They are traces of evolutionary history. Evolution rarely starts from scratch. Instead, it modifies existing structures, adjusting them to new functions over time. Older biological solutions remain embedded within newer ones. In this sense, the human body can be thought of as a layered record of evolutionary experimentation. Deep within our cells are genetic sequences inherited from ancient ancestors that lived hundreds of millions of years ago. Our vertebrate backbone reflects an ancient body plan shared with fish and reptiles. Our grasping hands echo the adaptations of primates that once lived primarily in trees. Even our brain, the organ most closely associated with human intelligence, evolved gradually from neural systems that existed long before humans appeared. When scientists study anatomy carefully, they often find clues pointing to these earlier stages of life's history. The arrangement of bones, the organization of nerves, the pathways of blood vessels, each reveals patterns that make the most sense when viewed through the lens of evolution. Seen this way, the human body is not simply a biological machine. It is more like an archive, storing information about ancient environments and evolutionary transitions. The curve of the spine hints at the moment our ancestors began walking upright. The structure of the hand tells a story about grasping branches and eventually shaping tools. The brain reflects the increasing importance of social cooperation, communication, and cultural learning. Each feature carries echoes of the past, and as scientists continue to examine fossils, genetics, and comparative anatomy, they uncover more details about the long journey that produced our species. To understand that journey fully, however, we must look further back than the appearance of humans themselves. Long before the first members of our species walked across the landscape, there existed earlier primates whose bodies already contained many of the structural foundations that would later shape the human form. The evolutionary roots of the human body begin not on open grasslands or in early human settlements, but high in the branches of ancient forests, where small mammals were beginning to develop the distinctive traits that would eventually define the primate lineage. Long before humans appeared, long before even the earliest apes moved through ancient forests, the foundations of the human body were already taking shape during a remarkable chapter in Earth's history. To understand where those foundations came from, we need to travel back to the beginning of the Cenozoic era. This era began about 66 million years ago, immediately after the extinction event that ended the age of the dinosaurs. A massive asteroid impact near what is now the Yucatan Peninsula triggered environmental changes that wiped out many large reptilian species. In the ecological space left behind, mammals began to diversify rapidly. Small mammals had already existed during the age of the dinosaurs, but they had mostly lived in the shadows, occupying limited ecological roles. Once the dinosaurs disappeared, those mammals suddenly found themselves in a world full of new opportunities. Over millions of years, mammalian species expanded into nearly every available environment. Some grew large and developed hooves for running across open plains. Others returned to the oceans and evolved into whales and dolphins. Still others adapted to life in the trees. It was within these forest environments that the earliest primates began to evolve. The ancestors of primates were likely small, agile mammals living in dense vegetation. Their survival depended on navigating a three-dimensional world of branches, leaves, and shifting light. In this environment, balance, precise movement, and accurate depth perception were essential. As natural selection acted over many generations, several distinctive anatomical features began to emerge. One of the most important was the development of grasping hands and feet. Instead of claws designed mainly for running or digging, early primates evolved flexible digits with flattened nails. These hands allowed them to grip branches securely while climbing or leaping through the forest canopy. Another defining feature was the position of the eyes. In many mammals, the eyes are placed on the sides of the head, allowing a wide field of view that helps detect predators. In primates, however, the eyes gradually shifted toward the front of the face. This forward-facing arrangement allowed the visual fields of both eyes to overlap. The brain could then combine the information from each eye to create stereoscopic vision. In other words, primates gained the ability to perceive depth with remarkable accuracy. If you have ever reached out to catch a falling object or carefully placed your foot while stepping over uneven ground, you have relied on the same visual ability that evolved in those ancient forest dwellers. Flexible shoulder joints also became a defining primate trait. The shoulder structure allowed a wide range of motion, enabling animals to swing, climb, and reposition themselves easily among branches. Over time, this mobility would later prove extremely important for tool use and complex hand movements in humans. Primatologists have spent decades studying these anatomical features in both living and fossil species. Among them is John Flegel, whose research on primate functional anatomy has helped clarify how skeletal structures relate to patterns of movement and ecological behavior. Flegel and other researchers examined how primates move through their environments, analyzing limb proportions, joint flexibility, and muscle attachments. Their work showed that primate bodies evolved in direct response to the demands of arboreal life. Another influential primatologist is Robert Sussman, who studied primate ecology and social behavior. Sussman emphasized how environmental complexity shaped primate evolution. Forest environments offered abundant food resources, but required careful navigation and coordination. In such settings, intelligence and sensory awareness became valuable survival tools. Navigating tangled branches while searching for fruit or insects required both physical agility and rapid decision-making. Over time, these pressures encouraged the development of larger brains relative to body size, a trend that would later continue dramatically in the human lineage. Yet the story of primate evolution has become even clearer with the rise of modern genetics. In recent decades, scientists have been able to compare the genomes of different species in extraordinary detail. By examining similarities and differences in DNA sequences, researchers can estimate how closely related species are and when they diverge from common ancestors. One of the most remarkable discoveries from this work is the close genetic relationship between humans and chimpanzees. The genomes of the two species are strikingly similar, sharing roughly 98 to 99% of their DNA sequence. This finding does not mean that humans evolved directly from modern chimpanzees. Instead, it indicates that both species share a common ancestor that lived several million years ago. Much of our understanding of these relationships has been transformed by the work of geneticist Svante Pääbo. Working at the Max Planck Institute for Evolutionary Anthropology in Germany, Pääbo helped pioneer the field of ancient DNA research. Extracting genetic material from ancient bones and fossils is extraordinarily challenging. DNA breaks down over time, and contamination from modern organisms can easily interfere with analysis. Despite these difficulties, Pääbo and his colleagues developed methods to recover and sequence genetic fragments from long-extinct human relatives. Their work revealed the genomes of Neanderthals and Denisovans, two ancient human lineages that once lived alongside early Homo sapiens. These discoveries showed that the evolutionary history of humans is more complex than previously imagined, involving periods of interbreeding between different hominin populations. Ancient DNA studies also reinforced the deep evolutionary connections linking humans with other primates. Many of the genes that guide the development of our bodies are shared across a wide range of species. This observation leads to another powerful concept in modern evolutionary biology known as deep homology. The term deep homology describes the phenomenon in which very different organisms rely on the same underlying genetic mechanisms to build their bodies. Evolutionary developmental biologist Sean B. Carroll has been one of the leading scientists explaining this idea. Carroll's research revealed that many genes controlling body structure evolved hundreds of millions of years ago. These genes are not unique to humans or even to mammals. Instead, they form part of a shared genetic toolkit used across the animal kingdom. For example, certain regulatory genes known as Hox genes control the layout of body segments during early development. These genes help determine where structures such as limbs, ribs, and vertebrae will form along the body axis. Remarkably, versions of these genes exist in creatures as different as fruit flies, fish, mice, and humans. This means that when evolution modifies body structure, it often does not invent entirely new genetic instructions. Instead, it adjusts how existing genes are used, when they are activated, and how strongly they are expressed. Through small modifications in developmental timing or gene regulation, evolution can produce significant anatomical changes. In this way, the basic blueprint of the human body is built upon genetic systems that originated long before the first primates appeared. Our hands, our vertebrae, our eyes, and even the structure of our nervous system reflect deep biological patterns inherited from distant ancestors. When scientists compare the anatomy of humans with that of other primates, they find countless shared features. The structure of the shoulder joint, the arrangement of teeth, the organization of the brain, and the pattern of muscles in the face all reveal common ancestry. These similarities are not coincidences. They are the result of evolutionary continuity. Yet despite these shared foundations, something unusual eventually happened within the primate lineage. Several million years ago, environmental conditions began to change in parts of Africa, where many early primates lived. Forests that had once dominated the landscape gradually gave way to more open habitats. Climate fluctuations altered rainfall patterns, vegetation, and food availability. As these environments shifted, the evolutionary path of one particular group of primates began to diverge from that of their closest relatives. Those changes would eventually lead to a lineage that walked upright, used tools, and developed the cognitive abilities that define our species today. But at this point in the story, those future transformations were still far away. The primate blueprint already existed, quietly carrying within it the anatomical possibilities that evolution would later shape into the human body. To understand why the human body evolved the way it did, it is not enough to look only at bones or genes. We must also look at the environments in which our ancestors lived. Evolution does not occur in isolation. It is always shaped by the conditions surrounding a species, by the climate, the landscape, the available food, and the unpredictable challenges of survival. For much of human evolutionary history, those conditions were far from stable. One of the scientists who has spent many years studying this relationship between environment and evolution is paleoanthropologist Rick Potts, director of the Smithsonian Human Origins Program in Washington. Potts and his colleagues have examined geological records, fossil evidence, and ancient climate data to understand the world in which early human ancestors lived. What their research revealed is that the landscapes of ancient Africa were not calm and predictable places where species could slowly adapt to one steady environment. Instead, they were locations of constant change. Rainfall patterns shifted repeatedly. Forests expanded and contracted. Grasslands appeared and disappeared. Lakes formed and later dried up. Entire ecosystems transformed over relatively short geological periods. This pattern of environmental fluctuation led Potts to propose an idea known as the variability selection hypothesis. The central principle of this hypothesis is that human evolution may have been driven not by a single type of environment, such as open grasslands or dense forests, but by the repeated instability of those environments. According to variability selection, species that survived were not necessarily the ones best adapted to a specific set of conditions. Instead, survival favored organisms capable of flexibility. Those that could adjust their behavior, diet, and movement as conditions changed had a better chance of enduring. In other words, adaptability itself became a key evolutionary advantage. Imagine living in a region where food sources might change dramatically over just a few generations. Fruit trees might dominate the landscape during one period, only to disappear when the climate becomes drier. Lakes rich with aquatic plants might form during wetter phases, then vanish as rainfall declines. A species specialized for only one type of food or habitat would struggle in such a setting. But a species capable of switching diets, exploring new territories, and developing different survival strategies would be more resilient. Over long stretches of time, this environmental unpredictability may have favored traits such as behavioral flexibility, problem-solving, and dietary diversity. To understand why the African landscape changed so frequently, scientists have looked closely at the geological processes shaping the region. One of the most important features influencing early human evolution is the East African Rift System. This immense geological structure stretches thousands of kilometers across eastern Africa. It formed as tectonic forces slowly pulled the African continent apart, along a series of deep fractures in the Earth's crust. As these tectonic plates gradually shifted, the land surface cracked and subsided in some areas while rising in others. This created valleys, lakes, volcanic highlands, and complex networks of rivers. Volcanic activity also played an important role. Layers of volcanic ash periodically settled across the landscape, preserving fossils and leaving behind geological markers that scientists can date with remarkable precision. These ash layers function almost like timestamps within the fossil record, allowing researchers to estimate when particular species lived. But tectonic activity also changed the physical environment in dramatic ways. Mountain ranges altered rainfall patterns by blocking moist air masses. Some regions became wetter while others grew drier. Lakes formed in newly created basins, only to shrink or disappear when climate conditions shifted. In some areas of East Africa, sediment cores taken from ancient lake beds show evidence of repeated cycles of expansion and contraction over hundreds of thousands of years. These cycles reveal that early hominin populations were living in landscapes that were constantly transforming. To better understand how these environmental changes affected ancient ecosystems, scientists have turned to chemical clues preserved in fossil remains. One of the researchers who made significant contributions in this area is geochemist Theo Curling from the University of Utah. Curling studied isotopes preserved in fossilized teeth and soil carbonates. Isotopes are slightly different forms of chemical elements that can act as environmental markers. By examining the ratios of certain carbon isotopes, scientists can determine the types of plants that dominated ancient landscapes. Plants using different photosynthetic pathways leave distinct isotopic signatures. Some plants, such as trees and shrubs common in forests, use a pathway known as C3 photosynthesis. Others, including many grasses found in open savannas, use a pathway called C4 photosynthesis. When animals eat those plants, the isotopic signatures become incorporated into their teeth and bones. By measuring these isotopes in fossil teeth, Curling and his colleagues were able to reconstruct ancient diets and ecosystems with surprising detail. Their research revealed that many early hominins lived during periods when African environments were shifting from dense woodlands toward more open grasslands. But these changes did not occur all at once. Instead, landscapes fluctuated repeatedly between forested and open habitats. This meant that early human ancestors likely experienced a mosaic of environments. In some regions they might encounter wooded areas rich with fruit and vegetation. In others they might find open terrain dominated by grasses and scattered trees. Such environmental diversity may have encouraged behavioral flexibility. A hominin capable of eating a wide range of foods would have a significant advantage in a variable landscape. Fruits, leaves, roots, seeds, insects, and eventually meat could all become part of the diet depending on what was available. Mobility also became increasingly important. In changing environments, food sources might appear in one location during a particular season, and then disappear the next. Individuals capable of travelling longer distances in search of resources would have a greater chance of survival. Over time, these pressures may have shaped both behaviour and anatomy. The ability to move efficiently across landscapes, to explore new habitats, and to adapt to changing ecological conditions gradually became more valuable. Variability selection suggests that human evolution was not simply about adapting to one particular environment. Instead, it was about developing the capacity to handle uncertainty itself. In a sense, our ancestors were evolving not only physical traits, but also behavioral strategies for dealing with unpredictability. Flexibility in diet, cooperation within social groups, and eventually the development of tools and cultural knowledge all likely contributed to this adaptive versatility. When viewed from this perspective, the human lineage begins to appear less like a straightforward progression toward a predetermined outcome, and more like a response to constant environmental challenges. Evolution was experimenting with different anatomical and behavioral solutions, guided by the shifting landscapes of ancient Africa. Within this dynamic world, certain physical traits gradually began to appear that would set the human lineage apart from other primates. One of the most significant of these traits involved a change in how our ancestors moved across the ground. As environments opened and travel distances increased, the ability to walk efficiently on two legs began to play an increasingly important role in survival. That anatomical shift would eventually reshape the structure of the spine, the pelvis, the legs, and even the muscles of the human body, marking the beginning of one of the most distinctive transformations in our evolutionary history. Among all the physical traits that distinguish humans from other primates, one stands out immediately. We walk upright. The ability to move across the landscape on two legs rather than four is known as bipedalism, and it is one of the defining characteristics of the human lineage. When scientists study fossil bones in order to determine whether an ancient species belongs to our evolutionary branch, the question they often ask first is simple. Did this creature walk on two legs? Bipedalism might seem ordinary to us because it is the way every human being moves through the world from early childhood onward. But from an evolutionary perspective, it represents a dramatic transformation in anatomy. Most mammals walk on four limbs. Even our closest living relatives among the primates, such as chimpanzees and gorillas, normally move using a form of locomotion known as knuckle walking. Their bodies are structured so that both the forelimbs and hind limbs support their weight as they move along the ground. Humans, by contrast, rely almost entirely on the hind limbs for support and movement. Our arms are freed from the primary task of locomotion, and instead become tools for carrying, manipulating, and creating. This change required extensive anatomical adjustments throughout the body. The spine, pelvis, legs, feet, and muscles all had to evolve in ways that would allow upright posture and efficient walking. Understanding when and how these changes began has been one of the central challenges of paleoanthropology. One of the most important discoveries in this area occurred in the year 1974 in the Afar region of Ethiopia. A team of researchers led by paleoanthropologist Donald Johanson uncovered a remarkably complete fossil skeleton belonging to an early hominin species known as Australopithecus afarensis. The skeleton was about 40% complete, an unusually high degree of preservation for a fossil of that age. Because the researchers had been listening to the Beatles' song Lucy in the Sky with Diamonds during their fieldwork, the fossil quickly became known simply as Lucy. Lucy lived roughly 3.2 million years ago. Although her brain was relatively small, closer in size to that of a chimpanzee than to a modern human, the structure of her skeleton revealed something extraordinary. Her pelvis was short and broad, shaped in a way that supports upright walking. The femur, or thigh bone, angled inward toward the knee, helping position the feet beneath the body's center of gravity. This inward angle is known as the valgus knee, a key feature of human bipedalism. Her spine also showed evidence of curvature that would have helped balance the upper body above the hips while standing and walking. Taken together, these anatomical features indicated that Australopithecus afarensis was capable of walking upright on two legs. Lucy did not walk exactly like a modern human. Her arms were relatively long, and certain features of her skeleton suggest she still spent time climbing in trees. But the fundamental shift toward bipedal locomotion had already begun. Another remarkable piece of evidence for early upright walking was discovered in Tanzania by the British paleoanthropologist Mary Leakey. In the 1970s, Leakey and her team were excavating a site called Latoli when they uncovered a set of ancient footprints preserved in volcanic ash. These footprints were created when a layer of ash from a nearby volcanic eruption settled on the ground, forming a soft surface. Soon afterward, a small group of hominins walked across the ash while it was still damp. Later, additional ash and sediment buried the footprints, preserving them for millions of years. The Latoli footprints date to roughly 3.6 million years ago. When scientists examined them carefully, they noticed something striking about their shape. The prints show clear heel strikes followed by the push of the big toe, just as occurs in modern human walking. The toes are aligned forward rather than splayed outward like those of apes. The pattern of pressure suggests a smooth transfer of weight from heel to toe during each step. In other words, the individuals who left those tracks were walking upright in a way that closely resembled human gait. Imagine the scene for a moment. Several small hominins moving across a quiet landscape shortly after a volcanic eruption, leaving footprints that would remain hidden beneath layers of sediment for more than 3 million years. Those footprints represent one of the earliest direct pieces of evidence showing that our ancestors had already adopted upright walking. But discovering that early hominins walked upright is only part of the story. Scientists also want to understand why this transformation occurred and how the human body supports this unusual form of movement. To explore those questions, researchers often combine fossil evidence with studies of modern human anatomy and biomechanics. One of the scientists who has contributed significantly to this field is Daniel Lieberman, an evolutionary biologist and paleoanthropologist at Harvard University. Lieberman studies how bones, muscles, and joints work together to produce movement. His research examines the mechanical stresses placed on the skeleton during activities such as walking and running. By measuring these forces, scientists can better understand which anatomical features are necessary for efficient upright locomotion. For example, when humans walk, the center of mass of the body moves slightly from side to side as weight shifts from one leg to the other. The pelvis plays a critical role in stabilizing this motion. Human pelvic bones are shorter and broader than those of apes. This shape allows muscles along the side of the hips to act as stabilizers, preventing the body from tipping too far when standing on one leg during a step. The spine also contributes to balance. Humans possess an S-shaped spinal curve, with gentle bends in both the lower back and the neck region. These curves help distribute weight and maintain the head above the pelvis while walking. Even the structure of the foot has evolved specifically for upright movement. In apes, the big toe functions almost like a thumb, allowing the foot to grasp branches while climbing. In humans, the big toe is aligned with the other toes and acts as a powerful lever during the final push of each step. The arch of the human foot also plays an important role. This arch acts like a spring, storing and releasing energy as the foot contacts the ground and pushes off again. When these anatomical features work together, they allow humans to walk long distances with remarkable energy efficiency. Yet evolutionary changes rarely occur because an organism suddenly decides to adopt an entirely new behavior. Instead, new functions often emerge from structures that originally served different purposes. This principle was described by evolutionary biologists Stephen Jay Gould and Elizabeth Verber, who introduced the concept of exaptation. Exaptation refers to a situation in which a biological trait evolves for one role and later becomes useful for another. Feathers in birds provide a classic example. Early feathers may have evolved primarily for insulation or display, but later they became essential for flight. In the case of human evolution, certain anatomical traits that once helped primates climb and balance in trees may have later been co-opted for upright walking on the ground. Long legs relative to arm length, flexible hip joints, and particular patterns of muscle attachment may have originally served one purpose before becoming useful in a different context. Evolution often works like a resourceful engineer, modifying existing components rather than inventing entirely new ones from scratch. Over many generations, natural selection refined these traits, gradually producing a body capable of stable and efficient bipedal movement. But upright walking did not simply affect how our ancestors moved across the landscape. It triggered a cascade of structural changes throughout the entire skeleton. Once the body began to balance vertically rather than horizontally, the spine, pelvis, rib cage, legs, and even the shape of the skull had to adjust. Those anatomical changes would continue unfolding over millions of years, gradually shaping the distinctive skeletal architecture that defines the human body today. Once upright walking began to appear among our early ancestors, the human skeleton entered a long period of gradual redesign. Standing and moving on two legs may sound simple, but from an anatomical perspective it is a surprisingly complex engineering challenge. When an animal moves on four limbs, its weight is distributed across a horizontal frame supported at several points. When that same body becomes upright, the entire structure must balance vertically over a much narrower base. Imagine stacking blocks into a tall column rather than spreading them out across the floor. The taller the stack becomes, the more carefully the pieces must align to keep everything balanced. In the human body, the skeleton performs that balancing act continuously. It holds the head above the spine above the pelvis, and the pelvis above the legs, all while allowing smooth movement and absorbing the forces produced by walking or running. To achieve this balance, several parts of the skeleton gradually change shape over millions of years. One of the most important changes occurred in the pelvis. In apes that move primarily on four limbs, the pelvis is long and narrow. This structure supports strong muscles used for climbing and for powerful movements of the hind limbs. In humans, the pelvis is shorter and broader. The bones curve outward to create a basin-shaped structure that supports the organs of the lower abdomen while also stabilizing the body during upright walking. When a person takes a step, the entire body briefly balances on one leg. Without special adaptations, the body would tend to tip sideways during this moment. Muscles attached to the sides of the pelvis prevent that tipping motion by pulling the pelvis level with each step. The shape of the pelvis therefore plays a crucial role in maintaining balance during bipedal locomotion. The spine also underwent significant transformation. In many four-legged mammals, the spine forms a gentle arch that runs horizontally from shoulders to hips. Because the body is supported by both front and hind limbs, the spine mainly serves as a bridge connecting those limbs. In humans, the spine supports the body vertically, which places different mechanical demands on its structure. The human spine forms what scientists describe as an S-shaped curve. The lower back curves inward, the upper back curves outward, and the neck curves inward again to position the head directly above the center of gravity. These curves act as shock absorbers. When we walk, each step generates forces that travel up through the skeleton. The curvature of the spine helps distribute those forces and prevents them from concentrating in a single location. Without these curves, upright walking would place enormous strain on the vertebrae and surrounding muscles. Another structural shift involved the rib cage. In many apes, the rib cage is shaped like a cone, wider at the bottom than at the top. This shape helps support powerful arm muscles used during climbing. In humans, the rib cage is more barrel-shaped. This change allows the upper body to rotate more easily during walking and running. As the legs move forward, the torso gently twists in the opposite direction, helping balance the movement of the body. Limb proportions also changed in important ways. Compared with other apes, humans have relatively long legs and shorter arms. Long legs increase stride length, making walking and running more energy efficient over long distances. Shorter arms reflect the reduced importance of climbing in everyday movement. The foot evolved into a highly specialized structure for supporting upright posture. The arch of the human foot distributes weight and stores elastic energy during each step. Meanwhile, the alignment of the big toe with the other toes allows the foot to function as a stable lever when pushing off the ground. All of these features work together to support the mechanics of upright movement. Scientists studying fossil bones have carefully examined how these skeletal features evolved over time. One researcher who made important contributions in this field is evolutionary anatomist Randall Sussman. Sussman studied the skeletal remains of early hominins and focused particularly on how the structure of the hands and feet revealed clues about movement and behavior. His research showed that many early hominins possessed a mixture of traits. Some features resembled those of modern humans adapted for walking, while others still reflected climbing abilities inherited from earlier primates. This mosaic pattern of anatomy suggests that evolutionary change often occurs gradually rather than all at once. Certain parts of the skeleton may shift toward a new function, while others retain older characteristics for many generations. Understanding why evolution proceeds in this gradual and sometimes uneven way requires exploring another important concept known as evolutionary constraint. Evolutionary developmental biologist Günther Wagner has studied how the structure of organisms can limit the directions evolution can take. An evolutionary constraint occurs when certain anatomical features cannot easily change because they are integrated into many other systems within the body. For example, the vertebrate spine must protect the spinal cord, support the head and upper body, and allow flexibility for movement. Any change to one aspect of the spine must also preserve these other functions. Because of these interconnected roles, evolution cannot simply redesign the spine from scratch. Instead, modifications occur slowly, adjusting existing structures rather than replacing them entirely. This explains why certain features of the human skeleton still reflect earlier stages of our evolutionary history. The basic arrangement of vertebrae, ribs, and limb bones follows a pattern inherited from ancient vertebrate ancestors that lived hundreds of millions of years ago. Even as evolution modifies specific shapes and proportions, the underlying framework remains recognizable. Another concept that helps explain skeletal evolution is modularity. Biologist Alessandro Minelli has explored how complex organisms are organized into modules, or semi-independent systems that can evolve somewhat separately from one another. In the skeleton, different regions such as the skull, spine, pelvis, and limbs can change at different rates. Evolution may alter the proportions of the legs without simultaneously changing the structure of the skull, or modify the pelvis without completely reshaping the rib cage. This modular organization allows evolution to experiment with new combinations of traits while maintaining overall functionality. Think of it as adjusting parts of a machine individually, rather than dismantling the entire mechanism at once. In early human evolution, modularity may have allowed features related to walking to develop gradually, while other aspects of the body continued performing older functions. The pelvis and legs began adapting for upright movement even while the arms retained some climbing ability. The spine developed curves supporting vertical posture, while the rib cage slowly changed shape to accommodate new patterns of breathing and movement. Over time, natural selection favored skeletal arrangements that made upright walking more efficient and stable. The result is the distinctive skeletal architecture seen in modern humans. Our skeleton balances height with mobility. It supports a relatively large brain, protects vital organs, and enables a wide range of movement from walking and running to climbing and throwing. Yet the human skeleton also reflects the compromises of its evolutionary past. The same spine that allows upright posture can be vulnerable to back pain because it evolved from an earlier structure designed for horizontal movement. The pelvis must balance two competing demands, supporting efficient walking while also allowing childbirth. These compromises are reminders that evolution works through modification rather than perfection. Despite these limitations, the skeletal system provides the structural framework for many of the capabilities that define human life. But bones alone do not move. Every step, every shift in posture, and every movement of the body depends on the coordinated action of muscles, tendons, and physiological systems that supply energy to those muscles. As the skeleton adapted for upright posture and long-distance travel, the muscular and metabolic systems of the human body also began to change in ways that would eventually allow our species to move across landscapes with remarkable endurance. When people think about human physical abilities, speed rarely comes to mind. Compared with many animals, humans are not especially fast. A cheetah can sprint faster than a sports car moving through city traffic. Even animals like antelope or horses easily outrun the fastest human sprinter. But speed is only one form of movement. There is another kind of athletic ability in which humans are surprisingly strong. It is not explosive speed, but endurance. When it comes to covering long distances over extended periods of time, the human body is remarkably capable. Many animals can sprint with extraordinary power, yet they tire quickly. Humans, on the other hand, can maintain a steady pace for long distances, sometimes for several hours at a time. In fact, among mammals of similar body size, humans are among the most efficient long-distance runners in the animal kingdom. This endurance ability is not an accident. It is the result of a series of evolutionary changes that gradually reshaped the human body. One of the most influential ideas explaining this transformation is known as the endurance running hypothesis. This theory was developed by evolutionary biologists Daniel Lieberman and Dennis Bramble in the early 21st century. Lieberman, working at Harvard University, and Bramble, a biologist at the University of Utah, examined the fossil record and modern human anatomy to identify features that might support sustained running. Their conclusion was striking. Many traits in the human body appear specifically suited for endurance running, even though other primates rarely run long distances in this way. For example, humans possess a large Achilles tendon that connects the calf muscles to the heel bone. This tendon acts like a powerful elastic spring. When the foot strikes the ground during running, the Achilles tendon stretches and stores mechanical energy. As the runner pushes off, the tendon releases that stored energy, helping propel the body forward. This spring-like action improves efficiency by reducing the amount of muscular energy required for each stride. Another important feature is the gluteus maximus, the large muscle that forms the bulk of the human buttocks. In other primates, this muscle is relatively small because their primary movements involve climbing or walking on four limbs. In humans, the gluteus maximus is unusually large and powerful. Research has shown that it becomes particularly active during running rather than walking. It stabilizes the trunk of the body and prevents the upper body from pitching forward with each step. While it may not be the most glamorous muscle in the body, it turns out to be one of the key contributors to human endurance locomotion. The structure of the neck also plays a role in running stability. Humans possess a ligament known as the nuchal ligament that helps stabilize the head during movement. This ligament acts like a tension cable, running along the back of the neck. As the body moves forward during running, the head naturally tends to bounce. The nuchal ligament reduces this motion, helping keep the head steady so that vision remains stable. Interestingly, this ligament is largely absent in other primates, but it appears in several animals adapted for sustained running, such as horses and dogs. Biomechanics research has helped scientists understand how these anatomical features work together during movement. Biologist David Carrier has studied locomotion in both animals and humans, examining how muscle activity and skeletal mechanics interact during running and walking. His research explores the physical forces involved in movement, including how energy is transferred through muscles, tendons, and bones. When humans run, the body behaves somewhat like a system of springs and levers. Tendons store elastic energy, muscles generate force, and the skeleton transmits that force into forward motion. The arch of the foot acts as a flexible spring. The Achilles tendon stretches and recoils. The long legs of humans increase stride length, allowing each step to cover more ground. Taken together, these adaptations make human running surprisingly efficient over long distances. But why would natural selection favor the ability to run long distances? One possible explanation involves a hunting strategy known as persistence hunting. Persistence hunting is a method in which hunters pursue an animal over long distances until the animal becomes exhausted. Instead of relying on sudden bursts of speed, the hunters maintain a steady pace while tracking the animal for several hours. Many mammals rely on rapid sprinting to escape predators, but they cannot sustain high speeds indefinitely. Humans, with their endurance running abilities, may have been able to keep moving long after other animals began to overheat or tire. Anthropologists studying traditional hunter-gatherer societies have documented persistence hunting in several cultures. One example comes from the San people of southern Africa, whose hunting techniques sometimes involve running animals such as antelope until the animals become exhausted. The human hunters move steadily across the landscape, following tracks and maintaining visual contact with their prey. Eventually, the animal slows down from fatigue or overheating, allowing the hunters to approach more closely. Persistence hunting requires not only endurance but also cooperation, navigation skills, and the ability to track animals across complex terrain. Even if this strategy was not the only hunting method used by early humans, the ability to move long distances would have provided important advantages. Endurance travel allows groups to explore larger territories, locate scattered food resources, and migrate across changing landscapes. The fossil record suggests that early members of the genus Homo began to expand their range across Africa and eventually into Eurasia. Such migrations required the ability to travel efficiently across open terrain. The evolution of endurance locomotion therefore may have supported both hunting strategies and long-distance movement across shifting environments. Yet sustained physical activity introduces another challenge. Muscles working continuously generate heat. Without an effective way to regulate body temperature, prolonged running would quickly lead to overheating. Animals that rely on sprinting often cool themselves through panting, which rapidly exchanges air and dissipates heat. However, panting becomes less effective during sustained running, because breathing must coordinate with movement. Humans solve this problem in a different way. Rather than relying primarily on panting, the human body evolved a sophisticated system for releasing heat through the skin. As muscles generate heat during activity, specialized sweat glands produce moisture on the surface of the skin. When that moisture evaporates, it carries heat away from the body. This evaporative cooling system allows humans to remain active even under warm conditions that would quickly exhaust many other animals. The evolution of endurance locomotion and the evolution of efficient cooling systems are closely connected. Running long distances under the sun requires a body capable of shedding heat effectively. Over time, the human body developed a unique combination of anatomical and physiological traits that support both sustained movement and temperature regulation. These changes prepared our ancestors not only to travel across open landscapes, but also to remain active during the hottest parts of the day when many predators were less active. The ability to move far, to track animals, and to remain active under challenging environmental conditions would gradually shape the lifestyle of early humans. But none of these endurance abilities would function without an equally important adaptation. A body capable of running for hours must also be capable of cooling itself efficiently, and that story leads directly to one of the most distinctive features of the human species, the remarkable system of skin and sweat that allows us to manage heat in ways few other mammals can match. Sustained movement across open landscapes introduces a challenge that every active body must solve. Muscles generate heat. In fact, most of the energy used during physical activity becomes heat rather than motion. If that heat cannot escape, body temperature begins to rise, and the risk of overheating quickly becomes dangerous. For animals that move quickly over short distances, this problem is manageable. A cheetah, for example, can sprint at astonishing speeds, but it cannot sustain that effort for long. After a brief chase, it must stop, pant heavily, and allow its body temperature to fall. Humans developed a different solution. Rather than relying primarily on short bursts of speed, the human body evolved systems that allow activity to continue for long periods without dangerous overheating. At the center of this ability lies one of the most distinctive features of human physiology, our remarkable system of sweating. Human skin contains millions of tiny structures known as eccrine sweat glands. These glands release a watery fluid directly onto the surface of the skin. As that moisture evaporates, it removes heat from the body. This process is known as evaporative cooling. Compared with most other mammals, humans possess an unusually high density of eccrine sweat glands. Estimates suggest that the human body contains between 2 million and 4 million of them distributed across the skin. Other mammals do sweat in limited ways, but they typically rely more heavily on panting to regulate temperature. Dogs, for example, cool themselves mainly through rapid breathing, which allows heat to escape through the moist surfaces of the mouth and respiratory system. Humans can pant as well, but our primary cooling mechanism is evaporation from the skin. This difference has profound consequences for endurance activity. When humans run or walk for long distances, sweating allows the body to release heat continuously, without requiring the breathing pattern to change dramatically. As long as sufficient water is available to replace lost fluids, sweating can maintain body temperature within a safe range even during prolonged exertion. But effective sweating requires another anatomical feature that makes humans somewhat unusual among mammals. Compared with our primate relatives, humans have relatively little body hair. Chimpanzees, gorillas, and most other mammals are covered with dense fur that provides insulation. While insulation can be beneficial in colder environments, it can also trap heat close to the body. The reduction of thick body hair in humans allows sweat to evaporate more efficiently from the surface of the skin. Air can move freely across the skin, carrying away heat as moisture evaporates. This combination of reduced body hair and extensive sweat glands forms a highly effective cooling system. The evolution of this system may have allowed early humans to remain active during the hottest parts of the day. Many large predators, such as lions and leopards, prefer to hunt during cooler periods at dawn or dusk. In contrast, humans equipped with efficient thermoregulation could travel and search for food under the midday sun. This advantage may have played a role in persistence hunting, where endurance and heat tolerance allow humans to pursue prey over long distances. Thermoregulation does not involve only sweating and hair reduction. Skin pigmentation also plays an important role in protecting the body from the effects of sunlight. One of the scientists who has studied this topic extensively is anthropologist Nina Jablonski. Her research focuses on the relationship between human skin color, ultraviolet radiation, and evolutionary adaptation. Ultraviolet radiation from the sun can have both harmful and beneficial effects on the body. On one hand, excessive ultraviolet exposure can damage DNA and increase the risk of skin cancer. On the other hand, ultraviolet light is necessary for the production of vitamin D, a nutrient essential for bone development and immune function. The challenge for evolving human populations was to balance these competing factors. Jablonski and her colleagues proposed that skin pigmentation evolved as a protective mechanism in regions with intense sunlight. Darker skin contains higher concentrations of melanin, a pigment that absorbs ultraviolet radiation and reduces damage to underlying tissues. Melanin also helps protect a nutrient known as folate, or vitamin B9, which plays an important role in DNA synthesis and fetal development. Ultraviolet radiation can break down folate circulating in the bloodstream, so higher melanin levels help safeguard this critical nutrient. In regions closer to the equator, where sunlight is strong year-round, darker skin pigmentation offers significant protective benefits. However, as human populations migrated into regions with lower levels of ultraviolet radiation, different selective pressures emerged. In areas with less intense sunlight, lighter skin pigmentation allows more ultraviolet radiation to penetrate the skin, helping the body produce sufficient vitamin D. Without enough vitamin D, individuals can develop skeletal problems such as rickets. Over thousands of years, natural selection favored different pigmentation levels depending on local environmental conditions. This variation in skin color across human populations illustrates a broader principle in evolution. Biological traits often represent adaptations to specific environmental challenges. The human skin is not simply a covering for the body. It is a complex organ involved in temperature regulation, protection from radiation, immune defense, and sensory perception. Its evolution reflects the climates and landscapes in which our ancestors lived. When early humans moved across open savannas under strong sunlight, effective thermoregulation became essential. Sweat glands allowed heat to escape. Reduced body hair improved evaporation. Skin pigmentation balanced the need for ultraviolet protection with the production of vital nutrients. Together, these adaptations allowed humans to remain active in environments that might overwhelm other mammals of similar size. Thermoregulation therefore became a key part of the broader evolutionary strategy that supported endurance movement and exploration. But maintaining activity in hot environments also requires energy. Muscles working continuously demand fuel, and the brain, which expanded dramatically during human evolution, requires even more. Meeting those energy demands required changes not only in anatomy but also in diet and metabolism. Over time, the foods our ancestors consumed began to shift, and those dietary changes would play a crucial role in shaping the next phase of human evolution. Every movement of the human body requires energy. Muscles contracting to lift a foot, lungs expanding to draw in air, neurons firing in the brain to process information. All of these processes rely on a constant supply of fuel delivered through metabolism. Metabolism is the collection of chemical reactions that convert food into usable energy. Without this continuous energy flow, even the most perfectly designed anatomy would remain motionless. As the human lineage evolved, shifts in diet played a powerful role in shaping the structure of the body. Changes in what our ancestors ate gradually influenced the size of the digestive system, the form of the teeth and jaws, and the balance of energy used by different organs. To understand why these changes occurred, it helps to remember that not all organs consume energy equally. Some tissues are metabolically expensive, meaning they require large amounts of energy to function. The brain is one of the most demanding organs in the body. Although it represents only about 2% of total body weight in modern humans, it consumes roughly 20% of the body's resting energy supply. Maintaining such an energy-hungry organ requires reliable access to high-quality food. One influential idea explaining how this energy demand could be supported is known as the expensive tissue hypothesis. This hypothesis was proposed by anthropologist Leslie A. Jello and evolutionary biologist Peter Wheeler in the year 1995. The central concept is based on a trade-off. If one organ system requires more energy, another system may need to use less in order to maintain overall balance. Aiello and Wheeler suggested that as the human brain grew larger during evolution, the digestive system became smaller and more efficient. Compared with many other primates, humans have a relatively short digestive tract. Our intestines are proportionally smaller than those of species that rely heavily on fibrous plant materials. Leaf-eating primates, such as colobus monkeys, possessed large digestive systems capable of fermenting tough plant fibers. These digestive systems require extensive gut tissue and complex microbial communities to break down cellulose. Humans, by contrast, consume foods that are generally easier to digest. A diet containing more nutrient-dense items such as meat, seeds, and cooked foods allows the digestive tract to be smaller while still providing sufficient energy. In this way, the energy saved by maintaining a smaller gut could be redirected towards supporting a larger brain. This trade-off does not mean the digestive system became unimportant. Rather, it suggests that the efficiency of digestion improved as dietary strategies evolved. Changes in diet are also visible in the fossil record through the structure of teeth and jaws. Early hominins often possessed large molars with thick enamel, suitable for grinding tough plant materials such as roots and seeds. Some species developed powerful jaw muscles capable of generating strong biting forces. Over time, however, the jaws of later human ancestors began to shrink. Teeth became smaller and more delicate, suggesting a shift toward foods that required less mechanical processing. Part of this change may have been driven by the increasing importance of animal protein in the diet. Meat provides concentrated calories and essential nutrients, including amino acids and fatty acids that support brain development. Evidence from archaeological sites shows that early members of the genus Homo were capable of butchering animal carcasses using stone tools. Cut marks found on fossilized animal bones reveal that early humans removed meat from carcasses and sometimes broke bones open to extract nutrient-rich marrow. But another important transformation in the human diet may have come from an unexpected source: fire. The idea that cooking played a major role in human evolution has been explored extensively by anthropologist Richard Wrangham of Harvard University. Wrangham proposed what is often called the cooking hypothesis. According to this idea, the controlled use of fire to cook food significantly increased the amount of energy humans could obtain from their meals. Cooking alters food in several important ways. Heat breaks down complex molecules in plants and meat, making them easier to digest. Starches become softer and more accessible to digestive enzymes. Proteins denature and unravel, allowing the body to absorb them more efficiently. Cooking also reduces the time and effort required for chewing. Raw tubers and tough plant materials can take hours to chew thoroughly, while cooked foods become softer and easier to process. Experiments comparing raw and cooked foods have shown that cooking can increase the number of calories the body absorbs from the same amount of food. This improvement in caloric efficiency would have provided a significant advantage for early humans with growing energy demands. Control of fire likely emerged gradually. Evidence from archaeological sites suggests that early humans may have been using fire regularly by around 400,000 years ago, though some researchers argue that controlled fire use may date back even further. Once cooking became common, it would have transformed daily life. Meals could be shared around firelight, food could be preserved through roasting, and previously inedible plants could become valuable sources of energy. Cooking also reduces harmful microbes and toxins in food, making it safer to consume. These dietary innovations allowed humans to obtain more energy while spending less time chewing and digesting food. But energy supply alone was not enough. The environments our ancestors inhabited were often unpredictable. Food availability could change with seasons, droughts, or shifting ecosystems. Under such conditions, survival favored metabolic flexibility. Metabolic flexibility refers to the body's ability to use different types of fuel depending on what is available. Humans can derive energy from carbohydrates, fats, and proteins. When one energy source becomes scarce, the body can adjust its metabolism to rely more heavily on another. For example, during periods when carbohydrate-rich foods are limited, the body can break down stored fat to produce energy. In extreme conditions, even certain proteins can be converted into glucose through metabolic pathways in the liver. This ability to switch between fuel sources allows humans to survive in a wide range of environments. Hunter-gatherer populations often experience fluctuations in food availability, yet their bodies are capable of adapting to these variations. Over evolutionary time, this metabolic flexibility may have helped early humans cope with the environmental instability that characterized many parts of ancient Africa. The combination of dietary diversity, improved food processing through cooking, and flexible metabolism created the energetic foundation needed for further evolutionary changes. Among those changes, one stands out as especially significant. The human brain began to expand dramatically. With increased energy available from improved diets and more efficient digestion, natural selection could support the growth of neural tissue. This expansion would transform the cognitive abilities of our species, influencing communication, social cooperation, problem-solving, and eventually the development of culture. But the story of the human brain is not simply about size. It is also about structure, organization, and the complex networks of neurons that allow humans to think, imagine, and communicate in ways unmatched by any other species on Earth. Among all the changes that occurred during human evolution, few are as striking as the transformation of the brain. Over millions of years, the brain of our ancestors expanded dramatically, eventually producing the organ that allows humans to reason, communicate, imagine, and reflect on their own existence. To appreciate the scale of this transformation, it helps to compare brain sizes across different species. Modern chimpanzees possess brains with a volume of roughly 400 cubic centimeters. Early members of the human lineage, such as Australopithecus, had brains that were only slightly larger, typically around 400 to 500 cubic centimeters. But when scientists examined fossils belonging to early members of the genus Homo, they notice a gradual but unmistakable increase in brain size. One of the earlier species in this genus is Homo habilis, which appeared around 2 million years ago. Fossils attributed to Homo habilis show brain volumes averaging about 600 cubic centimeters, noticeably larger than those of earlier hominins. Later species such as Homo erectus exhibited even larger brains, often reaching 900 cubic centimeters or more. By the time anatomically modern humans appeared roughly 300,000 years ago, the average brain volume had reached around 1300 to 1400 cubic centimeters. In other words, the human brain more than doubled in size over the course of our evolutionary history. But brain size alone does not fully explain human intelligence. What matters just as much is how the brain is organized and how different regions communicate with one another. The expansion of the brain was accompanied by changes in neural structure, connectivity, and function. One idea that attempts to explain why the brain expanded is known as the social brain hypothesis. This hypothesis was developed by evolutionary anthropologist Robin Dunbar at the University of Oxford. Dunbar proposed that increasing social complexity drove the evolution of larger brains. Living in social groups offers many advantages. Cooperation can improve hunting success, protect individuals from predators, and help raise offspring. But social life also introduces challenges. Individuals must recognize other members of their group, remember past interactions, and anticipate the behavior of others. Maintaining alliances, resolving conflicts, and communicating intentions all require cognitive effort. According to Dunbar, as primate social groups grew larger, individuals needed more mental capacity to manage these relationships. Dunbar examined the relationship between brain size and group size across different primate species. His analysis revealed a consistent pattern. Species with larger brains tended to live in larger and more complex social groups. When Dunbar applied this relationship to humans, he estimated that the average number of stable social relationships a person can maintain is roughly 150. This estimate became known as Dunbar's number. While the exact number may vary depending on circumstances, the idea highlights how human cognition may have evolved partly in response to the demands of social life. A larger brain allowed early humans to track relationships, cooperate effectively, and share information within groups. But expanding the brain introduced new biological challenges. As brain tissue grows larger, it produces more heat. Neural activity generates energy, and that energy must be dissipated to prevent overheating. Anthropologist Dean Falk proposed a theory addressing this issue known as the radiator theory. Falk suggested that certain changes in the vascular system of the skull may have helped cool the growing human brain. In particular, the network of blood vessels near the surface of the brain may have evolved to act as a kind of biological radiator. Blood flowing through these vessels could carry heat away from neural tissue, helping maintain stable temperatures. While scientists continue to debate the details of this theory, it highlights an important point. Evolutionary changes in one part of the body often require supporting changes in other systems. The expansion of the brain was not simply a matter of adding more neurons. It required adjustments in blood flow, metabolism, and skull structure to support the energetic demands of neural activity. At the same time, the organization of the brain itself was changing. Certain regions involved in language, planning, and abstract thought became more developed. Neural networks grew more interconnected, allowing information to flow between different parts of the brain with increasing efficiency. One scientist who has explored these changes in depth is Terence Deacon, a neuroscientist and anthropologist known for his research on the evolution of symbolic cognition. Deacon argued that the human brain evolved not just to process information, but to represent ideas symbolically. Symbolic thinking allows humans to assign meaning to sounds, gestures, and visual patterns. Words, for example, are not simply noises. They represent objects, actions, and concepts that exist in the world. This ability to create and interpret symbols forms the foundation of language. According to Deacon, language did not appear suddenly in a single evolutionary leap. Instead, it emerged gradually as brain structures supporting communication and symbolic reasoning became more complex. Early forms of vocal communication likely evolved first, allowing individuals to signal warnings, coordinate activities, or maintain social bonds. Over time, these signals may have developed into more structured systems of communication, eventually leading to the sophisticated languages spoken by humans today. Language dramatically expanded the power of human cooperation. Knowledge could be transmitted across generations, allowing cultural traditions to accumulate and spread. This process of cultural transmission accelerated human adaptation to changing environments. Yet even as the brain expanded and reorganized, the rest of the body continued to evolve alongside it. The skull had to enlarge to accommodate the growing brain. Changes in facial structure altered the shape of the head. Muscles of the jaw and face gradually shifted as dietary habits and communication methods evolved. These changes affected not only the mechanics of chewing and breathing, but also the ways humans express emotion and communicate with one another. Facial expressions, vocal sounds, and subtle movements of the mouth and eyes all contribute to the complex system of human social interaction. In this way, the evolution of the brain is closely linked to the evolution of the face and the structures that support communication. The growing human mind required new ways to convey thoughts, emotions, and intentions. As brain size increased and symbolic thinking emerged, the anatomy of the face and vocal system began to adapt in ways that would eventually support one of the most powerful tools humans possess: the ability to communicate through expressions, sounds, and eventually spoken language. As the human brain expanded over the course of evolution, the skull that housed it gradually changed shape. The growth of the brain did not occur in isolation. It altered the structure of the entire head, influencing the arrangement of bones, muscles, and soft tissues that form the human face. When scientists compare the skulls of early hominins with those of modern humans, they notice several important differences. One of the most obvious is the reduction in facial projection. In many early hominin species, the face extended forward from the skull in a pronounced muzzle-like shape. This forward extension is known as prognathism. It is common among apes and other primates, where the jaws and teeth play a major role in processing tough foods. Over time, the human face became flatter and more vertically aligned beneath the brain case. This shift occurred gradually as the skull expanded upward and backward to accommodate the growing brain. Paleoanthropologist Bernard Wood has spent much of his career studying these patterns of cranial evolution. Through careful analysis of fossil skulls from across Africa and Eurasia, Wood and other researchers have documented how the structure of the hominin head changed across millions of years. One consistent trend involves the shrinking of the jaws and the reduction of tooth size. Earlier hominins often possessed large molars with thick enamel and powerful jaw muscles suited for grinding fibrous plant materials. As diets diversified and cooking became more common, the need for extremely powerful chewing diminished. Over time, the muscles that once anchored large jaws became smaller, and the bones of the face gradually shifted into a more compact arrangement. The reduction of facial projection also allowed the skull to become more balanced above the spine. In species with projecting faces, the head's centre of mass lies farther forward. Flattening the face helps position the head more directly over the neck, improving balance in an upright posture. This may seem like a small detail, but small changes in the geometry of the skull can have important consequences for how the body carries and moves the head. Yet the human face is not simply a mechanical structure supporting chewing and breathing. It also serves as one of the most expressive surfaces in the animal kingdom. Beneath the skin of the human face lies a complex network of muscles that allow subtle movements of the eyebrows, eyelids, lips, and cheeks. These muscles enable an extraordinary range of facial expressions. A slight raising of the eyebrows can signal curiosity. A tightening around the mouth may convey uncertainty. A broad smile can communicate friendliness or reassurance. Many primates use facial expressions as part of their social communication, but humans appear to have developed especially refined control over these muscles. The evolutionary importance of facial expression becomes clearer when considering the role of communication in human societies. Humans are deeply social creatures. Cooperation within groups has been essential for survival, from sharing food to coordinating hunting and caring for offspring. Facial expressions provide rapid and often unconscious signals about emotional states and intentions. Long before spoken language developed fully, early humans may have relied heavily on visual cues from the face to understand one another. Anthropologists studying modern human populations have found that many basic facial expressions are recognized across cultures. Emotions such as happiness, surprise, anger, fear, and sadness can often be identified from facial movements alone. This suggests that the ability to produce and interpret facial expressions may have deep evolutionary roots. The muscles responsible for these expressions belong to a group known as the muscles of facial expression, which are connected directly to the skin rather than to other bones. This unusual arrangement allows the skin of the face to move in highly flexible ways. Over time, natural selection may have favored individuals capable of conveying emotional information clearly through facial cues. A well-timed smile might strengthen social bonds. A warning expression could alert others to danger. Even subtle changes in eye movement can communicate attention or suspicion. Facial anatomy therefore became closely linked with social behavior and group communication. The structure of the face also influences the way humans produce sounds. The position of the jaw, lips, and tongue all contribute to shaping the sounds that emerge from the mouth. These structures interact with airflow from the lungs and vibrations produced by the vocal cords. In early hominins with larger projecting jaws, the range of possible mouth shapes may have been more limited than in modern humans. As the face became flatter and the jaw smaller, the arrangement of the oral cavity changed. This created greater flexibility in shaping sounds with the tongue and lips. The lips themselves are especially important in human speech. They allow the production of sounds such as the ones heard in words, beginning with letters like B, P, and M. Subtle movements of the lips also help shape vowel sounds by altering the size and form of the mouth opening. These anatomical features gradually expanded the range of sounds humans could produce. The interaction between facial anatomy and communication illustrates a broader principle in evolution. Physical structures often serve multiple functions simultaneously. The human face supports chewing and breathing. It helps maintain balance of the skull. It expresses emotions and communicates social signals, and it contributes to the production of vocal sounds. Evolution rarely redesigns a structure for only one purpose. Instead, it modifies existing anatomy so that several functions can coexist within the same framework. In the case of humans, changes in facial structure created new opportunities for more complex communication. The flattening of the face, the reduction of jaw size, and the increased flexibility of facial muscles all contributed to a head capable not only of housing a larger brain but also of expressing and transmitting information with remarkable subtlety. But facial expressions alone cannot convey the full range of ideas humans exchange with one another. To share detailed thoughts, stories, instructions, and knowledge across generations, another evolutionary development became necessary. That development involved the transformation of the vocal system itself, leading to one of the most distinctive abilities in the entire animal kingdom: the capacity for structured speech. The human voice is one of the most versatile instruments found in nature. With a small set of anatomical structures working together, humans can produce thousands of distinct sounds, combine those sounds into words, and assemble those words into sentences capable of expressing ideas about the past, the future, or even imaginary worlds. This ability did not appear suddenly. It emerged gradually through a combination of anatomical changes and neurological developments that transformed the way early humans communicated. At the center of this transformation lies the vocal tract. The vocal tract is the system of organs that shapes sound as air moves from the lungs through the throat and out of the mouth or nose. Several key structures contribute to this process. Air pushed upward from the lungs passes through the larynx, often called the voice box. Inside the larynx are the vocal cords, thin folds of tissue that vibrate as air flows across them. These vibrations produce the basic sound of the human voice. That sound then travels through the throat, mouth, and nasal cavities, where its shape is modified by the tongue, lips, jaw, and soft palate. By adjusting these structures, humans can create the wide variety of sounds used in spoken language. The evolution of this system required several important anatomical changes. One of the most significant involved the position of the larynx. In many mammals, including most primates, the larynx sits relatively high in the throat. This position allows animals to breathe and swallow at the same time without choking. In humans, the larynx is positioned lower in the neck. This change increases the length of the vocal tract and creates a larger resonating chamber above the vocal cords. The longer vocal tract allows for a greater range of distinct sounds. Vowels, in particular, depend on subtle variations in the shape of the mouth and throat. A longer vocal tract makes it easier to produce clear distinctions between different vowel sounds. However, this anatomical arrangement comes with a trade-off. Because the human larynx sits lower in the throat, the pathways for breathing and swallowing intersect more closely. This increases the risk of choking compared with most other mammals. In other words, the ability to produce complex speech required accepting a small but real risk every time we eat. This may sound like an unusual evolutionary bargain, but it illustrates how strongly natural selection favored improved communication. One researcher who studied these anatomical changes extensively is linguist and evolutionary scientist Philip Lieberman. Lieberman analyzed how the structure of the vocal tract influences speech production. Through a combination of anatomical studies and computer modeling, Lieberman demonstrated that the human vocal tract is uniquely suited for producing the full range of sounds used in spoken language. He also examined fossil skulls to estimate how the vocal tracts of earlier hominins may have functioned. These studies suggest that species such as Homo erectus may have been capable of producing some speech sounds, though probably not the full range used by modern humans. Speech likely evolved gradually as both anatomy and neural control became more sophisticated. Another important piece of the puzzle involves genetics. In the early 2000s, researchers identified a gene that appears to play a significant role in speech and language development. This gene is known as FOXP2. The discovery emerged from studies of a British family in which several members across multiple generations experienced severe difficulties with speech and language. These individuals struggled to coordinate the precise movements required for clear articulation. Genetic analysis revealed that the affected family members carried a mutation in the FOXP2 gene. Geneticist Simon Fisher and his colleagues studied this gene in detail and discovered that it influences the development of neural circuits involved in motor control and speech production. The FOXP2 gene is not unique to humans. Versions of it exist in many animals, including birds and mice. However, the human version contains specific changes that appear to affect the way brain circuits involved in vocal control develop. These genetic modifications may have contributed to the fine motor coordination required for spoken language. Language did not emerge solely from changes in anatomy or genetics. It also depended on the development of complex neural networks within the brain. Regions such as Broca's area and Wernicke's area play important roles in language production and comprehension. These areas coordinate the planning of speech movements and the interpretation of spoken words. The growth and reorganization of these neural systems allowed early humans to process and generate increasingly complex sequences of sounds. Language therefore evolved through a combination of anatomical, genetic, and neurological changes that reinforced one another over time. As communication became more sophisticated, it transformed the way human groups functioned. Speech allowed individuals to share information about food sources, warn others about danger, and coordinate activities such as hunting or migration. Instructions could be passed from experienced members of a group to younger individuals, allowing knowledge to accumulate across generations. This process of cultural transmission became one of the defining features of human evolution. In many animal species, behaviors must be rediscovered by each generation through trial and error. Humans, by contrast, can learn from one another through language. A technique for making a tool, preparing food, or navigating a landscape can be taught verbally rather than discovered independently. Over time, this ability to transmit knowledge allowed human cultures to grow increasingly complex. Language also strengthened social bonds within groups. Storytelling, humor, and shared narratives helped reinforce group identity and cooperation. Through spoken language, humans could express ideas about events that were not immediately present. Plans for the future could be discussed. Memories of past experiences could be preserved. These abilities expanded the cognitive landscape of human societies. Yet language itself did not appear fully formed. It likely developed through many intermediate stages. Early vocal signals may have resembled the calls used by other primates to signal danger or maintain contact with group members. Gradually, these signals became more structured, allowing combinations of sounds to represent different meanings. As brain circuits evolved to process these patterns more efficiently, language grew more expressive. The anatomical features supporting speech, including the tongue, lips, and vocal tract, became finely tuned for producing precise sounds. But speech is only one half of communication. To create the tools, shelters, and technologies that define human culture, another set of evolutionary adaptations became equally important. Those adaptations involved the remarkable abilities of the human hand. If the human brain provided the capacity to imagine new possibilities, the human hand provided the means to bring those ideas into the physical world. Together they form one of the most powerful partnerships in the history of evolution. The human hand is an extraordinary piece of biological engineering. With its combination of strength, sensitivity, and precision, it allows us to perform an astonishing range of actions. We can grasp a heavy rock, thread a needle, sculpt clay, type a message, or gently pick up a fragile leaf without damaging it. This versatility depends on a particular anatomical feature known as the precision grip. The precision grip occurs when the tip of the thumb presses against the tips of one or more fingers, allowing extremely fine control of small objects. This grip is different from the power grip, which involves wrapping the fingers around an object such as a branch or a hammer. Many primates possess grasping hands suited for climbing, but the human hand is uniquely adapted for precision manipulation. The thumb is relatively long and highly mobile, capable of rotating across the palm to meet the fingertips. This movement is known as thumb opposition. Opposition allows the hand to stabilize small objects between the thumb and fingers, giving humans remarkable control over delicate movements. The bones of the hand also reveal subtle adaptations that support this capability. The joints of the fingers allow fine adjustments in position, while specialized muscles in the palm provide precise control of finger movements. But these capabilities did not appear suddenly. Evidence from fossils suggests that early hominins already possessed hands capable of some degree of tool manipulation. Over time, natural selection favored anatomical features that improved manual dexterity. Studying these ancient hands requires careful analysis of fossil bones. Researchers such as Matthew Skinner and Katerina Harvati have examined fossilized hand bones using advanced imaging techniques. Their work has revealed that certain early hominin species possessed finger bones, shaped in ways that supported powerful grips as well as precision manipulation. Some early species retained curved finger bones suited for climbing, while later members of the genus Homo displayed straighter fingers more consistent with regular tool use. These findings suggest that the evolution of the hand involved a gradual transition from climbing adaptations toward increased manipulation abilities. The story of human tool use becomes clearer when archaeologists examine the physical artifacts left behind by our ancestors. Stone tools provide some of the most direct evidence of early technological behaviour. One remarkable discovery related to early tools was made by archaeologist Sonia Harmond and her colleagues in the year 2015. Working at a site called Lomekwi in Kenya, Harmond's team uncovered a collection of stone tools dating back approximately 3.3 million years. These artifacts are among the oldest known tools ever discovered. The Lomekwi tools were created by striking stones together to produce sharp edges suitable for cutting or pounding. Although these tools were relatively simple, their existence suggests that early hominins had already begun to intentionally modify objects for specific purposes. Tool use likely began as a flexible behaviour rather than a fully developed technology. An early hominin might pick up a stone to crack open a nut or break bones to access marrow. Over time, individuals who learned how to shape stones more effectively gained advantages in obtaining food. These behaviours could then be observed and copied by others. In this way, tool-making became a cultural activity as well as a biological one. Cultural knowledge refers to information and behaviours that are learned socially rather than inherited genetically. Unlike genetic traits, cultural practices can spread rapidly within a population through teaching and imitation. Two researchers who explored the interaction between biology and culture are anthropologists Peter Richardson and Robert Boyd. Richardson and Boyd developed the concept known as gene culture coevolution. This theory proposes that cultural behaviours can influence the direction of biological evolution, while biological traits can in turn affect the development of culture. For example, the use of tools may have created selective pressure favouring individuals with greater manual dexterity and improved motor control. Over many generations, these traits could become more common in the population. At the same time, improved hands and brains would allow more sophisticated tools to emerge, further shaping cultural practices. This feedback loop between biology and culture helped accelerate human evolution. Once tool-making became established, it transformed many aspects of daily life. Stone tools allowed early humans to cut meat more efficiently, break bones to access nutritious marrow, and process plant materials that would otherwise be difficult to eat. Later innovations introduced more specialized tools such as hand axes, scrapers, and blades. Each new technology expanded the range of resources humans could exploit. Tool use also influenced cognitive development. Creating a tool requires planning, coordination, and an understanding of cause and effect. A stone must be struck at the right angle to produce a sharp edge. A wooden shaft must be shaped to attach a spear point securely. These activities require the brain to visualize outcomes and control complex sequences of movement. Over time, the demands of tool-making and tool use may have contributed to the expansion of neural networks involved in motor control and spatial reasoning. But hands do more than manipulate objects. They are also powerful sensory organs. The human hand contains thousands of specialized nerve endings that detect pressure, texture, vibration, and temperature. This sensory feedback allows the brain to adjust movements in real time. When a person holds a tool, the hand becomes an extension of the mind's interaction with the environment. Through the hand, humans explore the physical world, shaping materials into objects that carry both practical and symbolic meaning. The partnership between brain and hand therefore represents a key element of human evolution. Yet hands cannot function effectively without guidance from the senses. To shape a stone tool or track an animal across a landscape, early humans needed accurate information about their surroundings. Vision, hearing, and other sensory systems evolved alongside manual abilities, helping humans interpret the environment and respond with precision. These sensory adaptations would become another essential component of the evolutionary story written within the human body. To survive in a complex world, an organism must first be able to perceive it. Movement, intelligence, and communication all depend on the ability to gather information from the surrounding environment. In humans, this information arrives through a network of sensory systems that allow us to see, hear, touch, taste, and smell. These senses did not appear fully formed in our species. They evolved gradually over millions of years, shaped by the ecological challenges faced by early primates and later human ancestors. Among these senses, vision has played an especially important role in primate evolution. Many mammals rely heavily on smell to navigate their surroundings. Dogs, for example, possess an extraordinary sense of smell capable of detecting chemical traces invisible to human perception. Primates, however, followed a different evolutionary path. Early primates living in forest environments benefited from sharp visual abilities. Moving among branches requires precise judgment of distance. A miscalculated leap could result in a fall, which in a tall forest canopy would be an extremely unpleasant outcome. To solve this challenge, primates evolved forward, facing eyes that provide overlapping fields of vision. When the brain combines the slightly different images received from each eye, it produces stereoscopic vision, commonly known as depth perception. Depth perception allows the brain to estimate distances accurately and coordinate movements with remarkable precision. The human visual system retains this primate heritage. Our eyes sit near the front of the face, and a large portion of the brain is devoted to processing visual information. But vision in primates involves more than depth perception. It also includes an ability that many mammals lack: advanced color vision. Most mammals possess only two types of color-sensitive cells in the retina, which limits their ability to distinguish between certain colors. Many primates, including humans, possess three types of these cells, allowing a broader range of color perception. This system is known as trichromatic color vision. Anthropologist Nathaniel Dominy has studied how color vision may have influenced primate diets. His research suggests that the ability to distinguish subtle differences in color helped early primates locate ripe fruits and young leaves among dense foliage. Ripe fruits often display colors such as red or orange that contrast strongly with green leaves. A primate capable of detecting these color signals would be more efficient at locating nutritious food. Over many generations, natural selection may have favored individuals with improved color discrimination, gradually strengthening this visual capability. Vision also became important for social communication. Facial expressions, body language, and subtle gestures all depend on visual cues. In human societies, much information is transmitted without words through posture, eye contact, and movement. The eyes themselves contain specialized muscles that allow precise control of gaze direction. Humans can rapidly shift attention from one object to another, coordinating visual input with movements of the head and body. Yet vision is only one part of the sensory landscape. Hearing provides another channel through which humans gather information about their surroundings. The human ear is a complex structure consisting of three main regions: the outer ear, the middle ear, and the inner ear. Sound waves enter through the outer ear and travel down the ear canal until they reach the eardrum. Vibrations of the eardrum are transmitted through three tiny bones in the middle ear known as the malleus, incus, and stapes. These bones amplify the vibrations and pass them to the fluid-filled inner ear, where specialized sensory cells convert them into electrical signals that travel to the brain. This system allows humans to detect sounds across a wide range of frequencies. Anthropologist Rolf Quam has studied the evolution of hearing by examining fossilized ear bones from ancient hominins. Because the bones of the middle ear are extremely delicate, well-preserved fossils are rare, but when they are found, they provide valuable clues about auditory capabilities. Quam's research suggests that early members of the genus Homo possessed hearing abilities similar to those of modern humans in certain frequency ranges, particularly those associated with spoken language. This finding supports the idea that auditory adaptations for speech perception may have begun evolving before fully developed language appeared. Hearing also plays an important role in environmental awareness. Sound can reveal the presence of predators, approaching animals, or other members of a group. In dense vegetation where visibility may be limited, auditory cues often provide the first warning of nearby activity. Human hearing is particularly sensitive to frequencies produced by the human voice. This sensitivity helps individuals distinguish speech from background noise and detect subtle changes in tone or emotion. But sensory systems do not operate independently. The brain integrates information from multiple senses to construct a coherent picture of the environment. Vision, hearing, touch, and other sensory inputs combine to guide movement and decision-making. When a person walks through a forest, the eyes scan for obstacles, the ears detect distant sounds, and the sense of touch provides feedback through the feet and hands. The brain constantly compares and interprets these signals, allowing humans to respond quickly to changing conditions. As the human brain expanded, these sensory systems became increasingly integrated with cognitive processes. Visual information could be linked with memory, allowing individuals to recognize familiar landscapes or track animals across long distances. Auditory signals could be associated with meaning, forming the basis for spoken language. Through this integration of sensory perception and cognition, humans gained the ability to interpret complex environments and adapt their behavior accordingly. This growing awareness of the world around them helped early humans navigate unfamiliar territories, cooperate with others, and respond to ecological challenges. Yet while perception and intelligence allowed humans to interact with their environment more effectively, another aspect of biology played an equally important role in shaping the future of our species. The process through which humans reproduce, develop, and raise their young would introduce its own set of evolutionary challenges and adaptations, influencing the structure of the body and the rhythm of human life itself. Reproduction lies at the center of evolution. Every trait that appears in the human body exists because it was passed successfully from one generation to the next. Yet the process of producing and raising offspring is not merely a biological routine. In humans, reproduction is connected to some of the most unusual developmental patterns found among mammals. One of the most striking differences between humans and many other animals is the complexity of childbirth. Human infants are born with relatively large heads compared with the size of their bodies. This is largely the result of the expanding brain that evolved within the human lineage. As brain size increased over millions of years, the skulls of newborns also became larger. At the same time, the human pelvis had already evolved to support upright walking. The shape of the pelvis is influenced by the demands of bipedal locomotion, which requires a relatively narrow structure that stabilizes the body during each step. These two evolutionary pressures created an anatomical challenge. A large infant head must pass through a birth canal shaped by the requirements of upright walking. Navigating this passage requires a series of rotations as the infant moves through the pelvis during childbirth. Anthropologist Sherwood Washburn examined this problem in the middle of the 20th century and proposed an explanation known as the obstetric dilemma hypothesis. Washburn suggested that human childbirth represents a compromise between two competing evolutionary pressures. On one side is the advantage of a large brain, which supports intelligence, learning, and complex social behavior. On the other side is the need for a pelvis narrow enough to support efficient upright walking. According to Washburn's hypothesis, the size of the newborn brain is limited by the dimensions of the birth canal. If the brain were significantly larger at birth, the infant might not be able to pass safely through the pelvis. This constraint may explain why human babies are born in a relatively helpless state compared with many other mammals. A newborn foal can stand and walk within hours. Human infants, by contrast, require months before they can sit upright, and years before they become fully independent. At birth, the human brain is only partially developed. Much of its growth occurs after birth during infancy and childhood. For many years, the obstetric dilemma hypothesis was widely accepted as the primary explanation for this pattern. However, more recent research has introduced additional perspectives. Anthropologist Holly Dunsworth and her colleagues have proposed an alternative explanation focusing on metabolic limits rather than pelvic size alone. Their research suggests that pregnancy itself places enormous energy demands on the mother's body. During late pregnancy, the developing fetus requires large amounts of nutrients and oxygen to support rapid growth. The mother's metabolism must supply this energy while also maintaining her own bodily functions. Dunsworth's work indicates that human pregnancy may reach a metabolic ceiling near the end of gestation. In other words, the mother's body may simply be unable to provide enough energy to support continued fetal growth beyond a certain point. According to this view, babies are born when the energetic demands of pregnancy approach the maximum level that the mother's metabolism can sustain. The size of the pelvis still plays an important role in childbirth, but metabolic limits may also influence the timing of birth. These different perspectives highlight how complex evolutionary processes can produce outcomes shaped by multiple interacting factors. Regardless of the precise mechanism, the result is clear. Human infants enter the world at an early stage of development compared with many other mammals. This extended period of growth after birth leads to another distinctive feature of human evolution, an unusually long childhood. Biologists studying patterns of growth and reproduction across species often use a framework known as life history theory. Life history theory examines how organisms allocate time and energy across different stages of life, including growth, reproduction, and survival. Different species follow different life history strategies. Some animals mature quickly and reproduce early, producing many offspring with relatively little parental care. Others grow slowly, reproduce later in life, and invest significant resources in raising fewer offspring. Humans belong to the second category. Human children spend many years developing physically and mentally before reaching maturity. This extended childhood is rare among mammals. The long period of growth allows the human brain to continue developing throughout early life. Neural connections multiply rapidly during infancy and childhood as the brain processes experiences from the environment. Children learn language, social rules, cultural traditions, and practical skills during these years. They observe adults, imitate behaviors, and gradually acquire the knowledge needed to function within their communities. Anthropologists have suggested that this prolonged learning period played an important role in the evolution of human culture. Skills such as tool-making, hunting techniques, and food preparation require practice and instruction. Passing these skills from one generation to the next takes time. Extended childhood provides the opportunity for learning to occur within supportive social groups. In many human societies, child rearing involves not only parents, but also other relatives and community members. This cooperative system of care allows children to develop within a network of relationships. The evolutionary benefits of this arrangement are significant. A long childhood allows individuals to accumulate knowledge and experience before becoming responsible for raising the next generation. Cultural traditions can become increasingly complex as they are refined and transmitted over time. Human development therefore reflects a balance between biological growth and cultural learning. Infants begin life dependent on caregivers. Over the years, they gradually acquire the physical strength, cognitive abilities, and social understanding needed for adult life. This extended developmental journey is one of the reasons humans are capable of building societies, technologies, and traditions that span generations. But growth and development occur within an environment filled with potential challenges. From the moment of birth, the human body must interact with a world full of microorganisms, pathogens, and environmental hazards. To survive these challenges, another evolutionary system becomes essential. The immune system, which protects the body from disease, represents a powerful example of how human biology has adapted to the pressures of life on Earth. From the moment a human infant enters the world, it encounters an environment filled with microscopic life. Bacteria, viruses, fungi, and parasites exist almost everywhere. In the soil, in water, in the air, and even on the surfaces of our own bodies. Some of these organisms are harmless or even beneficial, while others have the potential to cause disease. To survive in such a world, the human body relies on a sophisticated defense system known as the immune system. The immune system is a network of cells, tissues, and molecular signals that identify and respond to foreign organisms. It operates through several layers of protection. The skin and mucous membranes provide physical barriers that prevent many pathogens from entering the body in the first place. If these barriers are breached, specialized immune cells detect the invaders and begin a coordinated response. Some of these cells attack pathogens directly, while others produce antibodies that recognize and neutralize specific microbes. Over time, the immune system can develop memory of past infections, allowing it to respond more quickly if the same pathogen appears again. Although this system may seem highly organized and precise, it did not arise through deliberate design. Like every other biological structure in the human body, the immune system evolved gradually through natural selection. Throughout human history, pathogens have exerted powerful evolutionary pressure. Diseases capable of spreading rapidly through populations can influence which genetic traits become more common over generations. In this sense, evolution can be thought of as an ongoing arms race between hosts and pathogens. Microbes evolve new strategies for infecting their hosts, while host organisms evolve defenses that reduce the impact of those infections. Genetic research has revealed several examples of how human populations adapted to specific diseases. One of the scientists who has contributed significantly to this field is geneticist Sarah Tishkoff, whose work focuses on the genetic diversity of African populations. Tishkoff and her colleagues have studied how certain genetic variations provide protection against diseases that have historically affected human communities. One well-known example involves malaria, a disease caused by parasites transmitted through the bites of infected mosquitoes. Malaria has shaped human evolution in regions where it has been common for thousands of years. Some genetic traits provide partial protection against severe malaria infection. The most famous of these traits involves the gene responsible for producing hemoglobin, the protein in red blood cells that carries oxygen through the bloodstream. A particular genetic variant of the hemoglobin gene produces a condition known as sickle cell trait. Individuals who inherit one copy of this variant from a parent often experience few symptoms, but they gain increased resistance to severe malaria. However, individuals who inherit 2 copies of the same variant develop sickle cell disease, a serious medical condition affecting the shape and function of red blood cells. This situation illustrates an important principle in evolutionary biology known as a genetic trade-off. A genetic trait that provides protection against one threat may carry disadvantages in other circumstances. In regions where malaria is common, the protective benefit of sickle cell trait helped maintain the gene in the population despite the risks associated with inheriting 2 copies. Such examples demonstrate how disease can shape the genetic landscape of human populations. Another field that explores these relationships between evolution and health is evolutionary medicine. Evolutionary medicine was pioneered by scientists such as Randolph Nesse and George C. Williams. Their work examines how understanding evolutionary processes can help explain patterns of disease in modern humans. Nesse and Williams argued that many medical conditions make more sense when viewed through an evolutionary lens. For instance, symptoms such as fever or inflammation are often unpleasant, but they may represent defensive responses that evolve to fight infection. Fever raises body temperature in ways that can slow the growth of certain pathogens. Similarly, inflammation increases blood flow to injured tissues, allowing immune cells to reach the affected area more quickly. From an evolutionary perspective, these responses may have been favored because they improved survival, even though they caused discomfort. Evolutionary medicine also explores how certain genetic adaptations helped humans survive in particular environments. One example involves lactose tolerance. In most mammals, the ability to digest lactose, the sugar found in milk, declines after infancy. The enzyme responsible for breaking down lactose becomes less active as the animal matures. However, in several human populations, genetic mutations allow the enzyme lactase to remain active throughout adulthood. This adaptation likely evolved in communities that practiced dairy farming, where milk became an important source of nutrition. Another example of genetic adaptation involves populations living at high altitudes. At high elevations, the air contains less oxygen than at sea level. Humans who move rapidly from low altitude to high altitude often experience symptoms such as fatigue, headaches, or shortness of breath. Yet populations that have lived for generations in high-altitude regions have developed genetic adaptations that help them cope with reduced oxygen levels. Research on Tibetan populations, for example, has identified genetic variations affecting how the body regulates hemoglobin production and oxygen transport. These adaptations allow individuals to function effectively in environments where oxygen availability is limited. Such examples illustrate the remarkable flexibility of the human genome. Over many generations, natural selection can favor genetic variations that improve survival in specific environments. However, these adaptations often come with trade-offs. A stronger immune response may help eliminate infections more quickly, but excessive immune activity can also contribute to autoimmune diseases in which the body attacks its own tissues. Similarly, genes that protect against certain diseases may increase susceptibility to others. Evolution does not produce perfect solutions. Instead, it produces compromises that balance benefits and risks under particular environmental conditions. As human societies changed, new challenges emerged. Agriculture brought people into closer contact with domesticated animals and with one another, allowing infectious diseases to spread more easily through dense populations. Later developments such as urbanization and global travel created even more opportunities for pathogens to move across the world. The evolutionary relationship between humans and disease therefore continues to unfold. Our immune systems carry traces of past encounters with microbes, and those encounters have shaped the genetic diversity seen in modern populations. But evolution does not stop simply because human societies become technologically advanced. The environments we live in today differ dramatically from those experienced by our ancestors, and these changes introduce new challenges for the human body. Understanding how ancient adaptations interact with modern lifestyles offers valuable insight into the health and biology of our species as it continues to evolve in a rapidly changing world. For most of human evolutionary history, our ancestors lived in environments that demanded constant physical activity, flexible diets, and close social cooperation. Survival often depended on walking long distances in search of food, adapting to unpredictable climates, and responding quickly to dangers in the surrounding landscape. The human body evolved under those conditions. Our muscles developed for endurance movement rather than long periods of stillness. Our metabolism became capable of storing energy during times of abundance and using it during periods of scarcity. Our brains evolved to navigate complex social relationships and to solve practical challenges in changing environments. But the environments in which many humans now live are very different from those faced by our ancestors. In evolutionary biology, this situation is known as evolutionary mismatch. Evolutionary mismatch occurs when traits that evolved under one set of conditions encounter a very different environment. Because biological evolution operates over long timescales, the human body may remain adapted to ancient circumstances even when the modern world changes rapidly. A simple example involves food. For most of human history, high-calorie foods were rare and valuable. Individuals who developed strong appetites for sweet or fatty foods had a survival advantage because those foods provided concentrated energy. In modern environments where calorie-rich foods are abundant and easily available, those same preferences can lead to excessive energy intake. The result is a mismatch between ancient metabolic adaptations and modern dietary conditions. Another example involves physical activity. Anthropologist Daniel Lieberman has studied how the human body evolved for movement and how modern lifestyles sometimes conflict with that design. Lieberman's research shows that early humans likely spent many hours each day walking, gathering food, and performing physically demanding tasks. The skeletal system, muscles, and cardiovascular system evolved to support this level of activity. Today, however, many people spend much of their time sitting. Modern technologies have reduced the need for physical exertion in everyday life. Transportation, machinery, and digital tools perform tasks that once required human labor. While these technologies provide many benefits, the reduction in daily movement can contribute to health conditions such as cardiovascular disease, obesity, and type 2 diabetes. Lieberman often points out that the human body is not designed for inactivity. It is built for regular movement. This does not mean that modern life is inherently harmful, but it does highlight how biological systems shaped by ancient environments sometimes respond differently when those environments change. Yet humans are not passive participants in their surroundings. Unlike most other species, humans actively modify the environments in which they live. This idea is central to a concept known as niche construction theory. Biologist Kevin Leyland and his colleagues developed this theory to describe how organisms can influence their own evolutionary conditions by altering their environment. Beavers provide a simple example. By building dams, beavers transform streams into ponds. This environmental change affects not only the beavers themselves, but also many other species that live in the same ecosystem. Humans engage in niche construction on an even larger scale. Agriculture reshaped landscapes by replacing wild vegetation with cultivated crops. Cities altered patterns of temperature, water flow, and animal populations. Modern technology has changed communication, transportation, and the availability of resources. Each of these changes creates new environmental conditions that influence human biology. For example, agriculture introduced new diets rich in grains and domesticated foods. Urban living increased population density, which affected the spread of infectious diseases. Advances in medicine reduced mortality from many illnesses that once exerted strong evolutionary pressure. These environmental transformations continue to shape human evolution. But genetic evolution is not the only mechanism through which biological change occurs. Another layer of complexity involves the field of epigenetics. Epigenetics studies how environmental factors can influence the way genes are expressed without altering the underlying DNA sequence. In other words, the instructions contained in genes can be activated or silenced depending on conditions experienced during development. Biologist Eva Jablonka has been one of the researchers exploring how epigenetic processes contribute to evolution and inheritance. Chemical markers attached to DNA molecules can regulate whether certain genes are active or inactive. These markers can be influenced by factors such as nutrition, stress, and environmental exposures. In some cases, epigenetic changes may persist across generations. This means that experiences affecting one generation could potentially influence the biological responses of the next. Although epigenetics does not replace traditional genetic inheritance, it adds another layer of complexity to the way organisms adapt to changing environments. The interplay between genes, environment, and culture continues to shape the human species. Modern humans are connected by global networks of communication and travel. Ideas, technologies, and cultural practices spread rapidly across continents. At the same time, genetic variation remains an important part of human diversity. Populations living in different environments continue to experience distinct selective pressures related to climate, diet, disease exposure, and lifestyle. Human evolution, therefore, did not stop with the appearance of Homo sapiens. It continues through the combined influences of biology, culture, and environment. The human body today carries traces of ancient adaptations shaped by millions of years of natural selection. At the same time, it exists within a world that is constantly being reshaped by human activity. Understanding this relationship helps explain both the strengths and the vulnerabilities of our species. Our bodies remain capable of remarkable endurance, creativity, and cooperation. Yet they also reflect compromises made under conditions very different from those many people experience today. Recognizing this evolutionary background allows us to view the human body not as a fixed design, but as a living system shaped by an ongoing dialogue between past and present. Every heartbeat, every breath, and every movement reflects the legacy of countless generations adapting to the challenges of survival. And that legacy continues to unfold as humans navigate the ever-changing landscapes of their own creation.